we're talking about digesting enzymes and finding the fingerprints. We got to base our first segment off of A, T, C, and G because that's the DNA we have to use. And from there, we're going to have to start cutting our DNA to figure out certain fingerprints we want to see. So let's go to our original problems from yesterday. To cut this, for number one, you guys figured out we have to cut DNA. All right, and to cut DNA, we need different enzymes. All right, and what was your answers for number one? From what organisms are restriction enzymes actually found in or derived in? Bacteria. No, yes, they are from bacteria, but now, what do they play in a natural host? What do usually these bacteria do in a natural host? I don't know, you tell me. Did anybody look this up? Alright, what did it say? Alright, so that's what you use for us. What are they doing in their natural host? They're like a protein. They do different jobs and functions throughout it. Now, we're going to look at two different ones in here. We have EcoRI and HIN3. That's how you call them. I think it's called Eco R1 and HIN3, or we call it HIN3 and EcoRI. How do we get these names? Where's EcoRI and HIN3 come from? Haley, what do we got? Yeah. E. coli, E, then coli is Ascocii, which is the bacteria that you guys don't want in your body on a normal basis because it will cause you harm. But E. coli, or EcoRI, is Eco R for the first segment. So it's a specific type one. But it comes from the bacteria E. coli, or E. coli. What about HIN3? Yeah. Does everybody else have that? Yeah. Super long one, and that's why we obviously break it down. But these are two bacteria that we're going to use as enzymes. And remember, enzymes are just proteins, and proteins have thousands of jobs. These proteins specifically cut in our body can do a couple things, which is, one, they can cut DNA, which is important for us when we look at DNA fingerprints. Now, if we go down here to number 3, every EcoRI has a specific, they're called target-specific areas. When you guys take medication, when you guys take flu medication, if you guys are taking alcohol, it always goes to a certain spot. It's going to have a target spot. Cancer drugs go to certain cells. Well, same with these enzymes. These enzymes only go to certain segments of the DNA. And depending on what it is, it only goes to that section. For this PSTL, or pistol it's called, we have this section, CTGCAG. It recognizes only that sequence. It doesn't matter how many other millions of base pairs, it recognizes that sequence and then it literally cuts it. Now, if we go back to our board presentation, PSTW, RFC we have up here already. It gets cut in this direction, that's on your paper. But then the next question talks about how does the enzymes EcoRI and HIN3 actually get cut. First off, we have to know the sequence. What is the sequence where it gets starting cut? Well, you just got to start reading the DNA strand. And the thing is, it's only unique. And the only unique thing for E. coli or EcoRI is this segment right here. When you're reading it, you have to read it so it reads GAATC, TTC, and CTTAAG. All right? Those are the main sequences you guys have to follow here. I was gonna this minute. Ah. So GAATTC and CTTAAG. That's the sequence you guys have to look for. Once you find that sequence, you have to cut it. And Eco-Rye cuts that very specifically. It cuts it. The G gets cut away from AATC, TTC, and CTTAAG. It gets cut down there. And it looks like we were to take this guy. Cut down the G, go over, and cut down the G. So if we blow it up a little bit, so we have G, whoops, magic pen, G, A, G, A, T, T, C, and C, T, T, A, G. Oh my gosh, seriously. This is a rough lesson for those juniors. We're having a rough time right now. Please stay up here. And I can't even write this. Backwards. So if we have the DNA segment, G, A, T, T, C, C, T, T, A, G. That is what our enzyme Eco-Rye is looking for. And what happens? It literally takes its scissors, cuts it, so it gets separated. Now, since this is not a straight down cut, there are some that we read that have blunt ends. So if you have, for a pistol or some other ones, you would have a random segment, CAATT. This would be GTTA. Some enzymes cut it right down the middle, and then when they separate it, it's CGAT and ATTA. And when it happens, when there's a clean cut, so right here there's no fragments, it's just 2 and 2, they match up. These are called blunt ends. If you don't have this on your paper, I would definitely put it. So anytime it cuts right down the middle and doesn't leave any fragments from one side to another, these are called blunt ends. Now, the other hand, HIN3 and Eco-Rye, they actually don't have blunt ends. And as you guys can see right up here when we did the dotted lines, it didn't go straight down, it cut across and made this zigzag kind of formation. And if we're going to rewrite it, so when we separated it, G's over here, we got C, T, T, A. The other segment would read A, T, T, C, and the G would be down here. So it's kind of cut jaggedly. Now, we call these, when they don't directly have a base pair down from each other, these are called sticky ends. Now, is there advantage to this? Not at all. If it's sticky ends or blunt ends, it's not going to change how we write it. It's not going to change how it goes in gel electrophoresis. But they just have specific names for sticky ends and blunt ends. Oh, I got the Y. Sticky ends and blunt ends. Now, once we find our DNA segments, then we have to do some math, and it's just adding or subtracting and counting. So stuff that we do in like 3rd and 4th grade, or maybe in 1st. So we're going to take our example from your paper. There's 2 examples, person 1 and person 2. And then there's questions. Person 1, you have to cut it up And then you have to talk about these restriction fragments. And then you have to talk about restricted fragments as in what is the size in BP. So we're going to go through here and we're going to figure out, we have to cut person one according to eco-ride. So we're just going to go across and we're going to try to find the segments GAATTC. If we find that, then we can cut it. So we got to go across. Our first letter we have to find is a G, then double T. So we have G, nope. All right, the second one in here. We got GAATTC, nope. That's an A, that doesn't count. Keep going, G, nope. GAATTC, there we go. So we cut it. So that's cut. So we've got one cut in here. And we just keep going. TTAT, no, GAATT. Ooh, another one. So we got that cut. Then we just keep rolling down. We're just trying to match it up. So I think we have GAATTC. We cut there. So now we've cut this 3 different times. So we have a specific fingerprint for this. So on this segment of DNA, on this person, because person's DNA is different, and depending who it is, we're going to have different cuts. And for this person's cut, this is going to be their fingerprint for this section of it. So then we've got to figure out number of restriction fragments, as in how many are there on here. So we've got 1 here, 2 here, this is number 3, and this is number 4. So we've segmented it into 4 different fragments. Now we've got to figure out, once we have these fragments, let's kind of have to see how big are these. Now we don't count them in mass, we don't have grams or kilograms. We start with what we started earlier today. We talk about A's, T's, C's, and G's, and we talk about base pairs. So how many of these base pairs are lined up with each other? Now, it has to be a pair. It can't be a sticky end is not going to count. I'm going to tell you guys that in a second, how that works. So, if we have this section, this first number 1, G, G, A, A, T, T, A, A, G, C, T, T, A, T, T, G. And then on the bottom, we have this extra T, T, A, A. Well, when we count, the specific word is base pair. So these sticky ends don't count because they're not in a pair. The only thing you count is how many are paired up. So we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Did I count that right, everybody? Is that 16? So we literally just counted. This is base pair 1, 2, 3, 4. And we got all the way down, and that's how many columns, if you want to think of it as columns, that's how many full columns we have on this base pair. And we have to have this paired. This TTAA is not paired up. And that's where the sticky part of it comes from. It doesn't have anything to stick to it, so it's actually out there and it has a little stickiness to it. So these don't count as base pairs. They don't. Everything before that, that's segmented, it is. So number 1, we have 16 BPs. Now I need you guys right now to go through segment 2, segment 3, segment 4, or fragment 2, 3, and 4. Count how many base pairs you have, and write them where I have the number 1 at. And I'll stop this recording so you guys can catch up as well. All right, and we're back. So segment 2, How many base pairs do we have for segment 2? 7? Do we create with 7, ladies and gentlemen? So we got 7 base pairs for that. Now remember, we don't count the sticky ends. The only ones we count, and if you guys are like, oh, it looks so much longer. Well, it really isn't, because if we just use a highlighter right here, that's 1, 2, 3, 4, 5, 6, 7. The 2 ends right there, as you guys can see, there's only 7 that have mates, are paired up. Everything else, the 2 sticky ends, yes, great. They're a big part, but they don't matter to us because they're not going to overall affect about how big it is. So, who's got segment 3? We'll go, Emily. How many do you have for segment 3 or fragment 3? Yay or nay, do we agree or disagree with her? Agree. So yeah, we'll go through there again, and not to make this long, but I want to make sure everybody sees this. We just go through and match them all the way up, all the way to here, and everything in between here and here count as base pairs. So finally we get to the last one. Why that doesn't make an arrow, I don't know. Segment 4, Brendan, what do we got? 5 base pairs. Good. So at this point we can go through and answer the 2 questions up here. First off, the number of restriction fragments, pieces of DNA after digestion. How many is there, everybody? 4. And then we have to, the size of the restriction fragments in base pairs from largest to smallest. So if we do this using our number line, what do we have first? 17, then 16, then 7, and finally 5. So those are the 2 important pieces of the puzzle for gel electrophoresis. So after we put the enzyme with the DNA, it digests the DNA, and it gives us this very specific 4 fragmented pieces of DNA. It gets eco-rye, digested it in that way. And we get fragments that are 17, 16, 7, and 5 base pairs big. Now that's going to be very important to us when we do gel electrophoresis. We're all kind of building this to make our fingerprint. And we're halfway there to seeing our fingerprint. So right now I want you guys to work in pairs or individually. And do person 2. Same thing. Find the fragments. We're still using eco-rye. So find the number of fragments, then go from largest to smallest, as in how many base pairs they take up. Yeah, we're back again. So I need 3 people. 1 to cut it, 1 to tell me how many fragments, and 1 to tell me how big the fragments are. Jacob, I don't think you've gone. So you can get up there and digest it for us, because that's a specific name, how we do it. We digest it. Lindsay, you're going to tell me how many fragments it is. And then, Ellie, I don't think you've gone yet. You can tell me how many base pairs this is. All right. Jacob, come on up, man. She's beating you to the punch already. So hopefully you've got 3 cuts in here, which means 4 fragments. That's fine. It's a learning curve. Look at the precision there. We've got 1 more cut left. Boom, that's how it's done. 4 segments. All right, Ellie, come on up and write from largest to smallest. Doesn't matter. Either way, I know. We'll just put it under the actual size of fragments, right on that line. Do we have a correlation between her numbers and your numbers? Awesome. Now, in the end, it doesn't matter where these segments come from. The biggest thing is the 8, 10, 12, and the 13. Those are the numbers we care about. Because this is going to be what we see on the gel electrophoresis. So this is going to be part 1. We review on this on Friday. We're going to get DNA. We're going to put enzymes on here, and we're going to put it in a hot water bath at our human temperature, 37 degrees Celsius. And the enzymes are literally going to go through the real DNA we have, and they're going to segment everything out for us. It's only going to take them about 35 minutes. These natural enzymes are going to break up the DNA we have, and it's going to make these segments. So there's no longer going to be one big strand of DNA. There's going to be all these segments all over. Then on Monday, we've got to take these DNA segments, these RLFPSs, and figure out, hey, if we put them through electricity, through a gel, what is it going to look like visually? Because obviously looking at this under a microscope, we can't see it because they're so small. So we need to know what this fingerprint looks like for person 1 and person 2 under their actual sizes. So that's where this comes into factor. So in part B right here, gel electrophoresis for restriction fragments. So this is our gel. It's going to be made out of agarose, which is the same thing as you guys made your gel out of in biology with agar. Now, all the time when we run a gel, it's called literally running because it runs down the gel. Now, always you have to have a standard marker. A standard marker is the first well, it's called. This first empty box is called a well, and when we run it, it gives us a standard marker. If this does not show up during our run, it means something went wrong and went awry. So what happens with person 1 and person 2 is exactly what we just did, but it's visual compared to numerical. So person 1, we're going to put their DNA in here, and we know from person 1 has 4 segments. We've got a 16, a 17, a 7, and a 5. Now, we know numbers. All we now have to correlate is numbers to make a visual picture of what's going on in this. So we're officially making the fingerprint now. Keyboard's killing me today. So this will be fine right here. Same concept. You guys have one that's half the size. Now, all we have to do, since there's standard markers, now we know where it's going to be at. This is going to be this first one that says 30. That is a segment that should have 30 base pairs. And then we have right here is 25, 20, 10, 5, and 2. Now you just go down person 1 and find approximately where 17 would be, where 16 would be, where 7 would be, and where I think 5 would be. So you're just making lines under person 1, You're making lines of approximately where it is. So just like they have lines for 20, make a line where there's a 17, a 16, a 7, and a 5. So that's your goal right now, and we'll come back to it in a second. So just to finish this up really quick, we should have, person A should have a line right here, person 1 here, and it's 7 and what else, guys? 7, 5. Person 2 was at what, everybody? 13, 12, 10, 8. So that's the DNA fingerprint for person 1 and A. So when we talk about fingerprints in DNA, that's what it is. So have an enjoyable afternoon, and I'll get you guys your homework later.